Okay, I'm gonna go back to the beginning. Yeah, so Kim Shah says, which ones are Russian? What specifically about Russian bees? Maybe I described those already. Tom says, is the aggressive trait reduced as they mate with other apis? Yeah, potentially. And so, I mean, if you have really aggressive colonies, they're mating with drones from Italian bees, for example, which be more docile, they tend to be less aggressive. Okay, but it can definitely work the other way around, where if you have less aggressive bees, like Italian honeybees, they're mating with drones from, Africanized honeybees, they can be more aggressive. And so males make a huge difference on how aggressive your colonies are going to be. And so I do know if you are part of a breeding program, and so this is one problem with a lot of these breeding programs, like the hygienic ones, like VSH. When you select for grooming behavior, hygienic behavior, the traits tend to be really tied with aggressive behavior. So it's really hard to separate aggressiveness with these hygienic traits. And so a lot of times, especially with pole line and the mid hygienic, they had really great bees, but they tend to be more aggressive. So they've had to take a lot more time to separate those traits together.
So do you, this is Kim, by the way, Garrett. I was asking earlier on the Russians which group they fell in, whether it was O or I. I couldn't remember all the subgroups, but um, if you're trying to start breeding your own queens, say you set up a predominantly drone yard a little further away from your apiary, do you want those drones to be calm and less hygienic and your queens be more hygienic and more aggressive, or vice versa?
Yeah, so which lineage do they come from? I guess they're sort of mutts right now, but I'd say primarily the M lineage, I believe. I'd have
Mm-hmm.
to look into what their background is, but I think they're mostly M lineage now, like Italians and Carniolans, a mixture of those two. As for so you're saying your queens, like do you want your queens to be more aggressive or your drones or vice versa? Is that kind of your question?
Right. Like, say you
Yeah.
wanted to start breeding queens and you were trying to get that
Yeah.
VSH, but also bring down the temperament, and not be too aggressive. Do you want to focus on
Yeah.
drones that are calm, from calm hives, or vice versa?
That's a really great question. So I would probably focus on the queens, and I really say that because you can't control who your queens mate with. So you could set up a bunch of drone producing colonies, but you can't guarantee that your queens are gonna mate with those drones. If you were running a closed breeding program, for example, I'd probably focus on the drone part of it. But just because you can't control where the queens are mating with, and I guess you might be isolated, for example, so maybe you can control it. But if you can't, I would just focus on the queens and select on the queen genetics that way.
Mhm.
So,
And
so, so,
then, do
le
you have any
sure.
pictures of the different types of breeds? Like, are the black queens usually German or Russian, and the golden, like some of them are real
Yeah.
yellow or real orange or real black. So can you show us the difference? Because I had somebody say, "Oh, you have buckfast bees," and I did purchase
Yeah.
buckfast bees at one point in time. But how do you know that they're buckfast just by looking at them?
Yeah, I have some pictures. Let me pull up the presentation again. These are kind of common pictures of them, but I will say that, I've sequenced bees that are really golden color that look Italian and they have more carniola in them. So sometimes it's hard to tell by color. And so you can have carniolan honeybees that are kind of goldish color. Now, typically the Italians are the lighter colors and the carniolans are darker colors, but oftentimes it's really hard to tell them apart. But let me share this quick. So this is a really great example of like an Italian honeybee, kind of lightish, goldish color. But if she's mating with a bunch of males, for example, that some are Carniolan background, you could have a mixture of like really light workers and some darker workers. So it kind of depends on who she's mating with. Um, see Africanized honeybees. Where's a picture of that? And usually the Caucasian honeybees are kind of like this color. This is what kind of Caucasian honeybees look like, darker. Carniolans are a little similar to that. Let me see if I find it. This is kind of example of a dark German bee, so they tend to be really, really dark, like a really black color without the stripation on the backs.
Mhm.
Whereas, let's see. Cyprian. So I'm kind of getting oriented here first. This is what a kind of a carniolans typically look like. They're a little bit darker, but they have kind of more of the yellowish hair on their thorax. They also tend to be a little bit bigger. So I'd say in general, if you're using colors, that the darker ones tend to be more carniolan, more Russian, whereas the lighter ones are more that kind of pole lines are more lighter color like Italians. Italians are more lighter colors. So, but I will say you really can't depend on the color because sometimes they can be darkish coloring. They can be a lot more Italian. So it's tough to tell them apart. Did that answer your question? I hope it
Yeah,
did.
yeah, that's very
Okay.
helpful. And then a buckfast bee. What is a buck? How can somebody tell what a buckfast looks like? Are they smaller, bigger, or
That's a good question. So I've never actually worked with Buckfast. I believe they're darker. I believe they're darker and similar to the Saskatraz, but
Mhm.
I'm not quite sure, and I really don't think you can tell them apart unless you do genetic testing. I mean, even with Africanization, like this is the Africanized honeybee. It's quite a bit smaller in yellowish color, but they're basically all mutts at this point, and so it's hard to tell whether they're like a Buckfast or Italian or Africanized bee because they're all mutts from different genetics. And so I don't think you can tell whether it's a Buckfast or not unless you do some sequencing. But that's not to say you can't. So maybe they do have like a maintained Buckfast line.
yeah, I don't know. I had a mentor tell me, oh, these are buckfast bees. I can tell by looking at them. I'm like, oh, well,
I
okay.
don't think so. I
I
I
can't
I
tell.
don't think so. No, I can't tell either. I can't tell either. So, sometimes yeah, I'll sequence a bean. I think like it's kind of like dogs where you do genetic testing, like, oh yeah, this is a terrier and this is this type of dog. And then we actually do sequencing. Oh yeah, it has a little bit of this other breed in it. So yeah, you can't always tell by just doing a visual test. Where's the Buckfast from, by the way?
I got those from Bee Weaver.
Okay, so
But that was my first year, and I lost both of those hives. One absconded, and the other, I think they both absconded actually. So, and then since then, I've been buying Italians, and I have put an order in for a Russian nuke.
okay, from where? I think there's a few
Oakley
producers here.
Apiaries, north of
Oh yeah.
Waco, New Elm.
Yeah, I think the Oakleys actually get it from the Coys, an actual breeding program. So yeah, that's a really I'd give it a shot. I've heard they're more aggressive, but I've worked with them and I haven't had any issues. So I've always liked the Russian bees.
Yeah, but I don't mind the aggression too much, but
Yeah. Well, and when they're aggressive, right, they tend to be more mite resistant too. And so there's some cost and benefits of both. And if there's a little aggressive and if you don't mind it, then it's a good route to take.
as long as they don't chase me around the yard, that's the main thing.
Exactly. Exactly. You
If
you
they
can
start
always
doing
requeen
that, then
them.
I'll start breeding them with my Italians.
Yep,
So
exactly. Well, Lee had a question. So, what does old lineage mean? And I actually wasn't sure. I think these are arbitrary letters, but I looked it up, and I think it stands for Oriental. I believe. So don't quote me on that, but that's maybe what the O comes from. But I'm not sure. I've actually never thought about that before. I've always just called them O, M, and C. So Lee had a question. Are you saying that before bees were imported, there were no honeybees in the United States? That's a good question. So I talked about this last week, and there may have been honeybees beforehand, but especially in Texas, I've been writing this chapter on the history of beekeeping in Texas, and there's no records that the Native Americans that were native to Texas maintained any bees. They didn't start beekeeping until around 1834 with the Austin colonies. Now, that's to say that bees weren't here beforehand because there's a lot of evidence that the Mayans were taking care of bees really a long time ago, and so bees could have been at least in the southern part of Texas, but I'm not quite sure. We really don't know that, but if we do some sequencing, maybe we can get sort of a better idea of when they were first came to the United States and when beekeepers are managing them. Yeah, and Kim asked, so where do buckfasts fall into? Yeah, I'd say they're kind of mutts. I'd say they're more that I mean they're probably mostly C lineage, mostly Italian Carniolans, maybe a little bit of Mellifera. Um, I think I did sequence them.
mm. I
I
did
did sequence
find
them, yeah.
that when they requeened pretty spicy. So I don't know if that was because of my drone population nearby, but I've talked to
Yeah.
other beekeepers in Texas that said that when their buckfast requeen, they were pretty hot.
Yeah, and it could be the drones because you're located near Huntsville, right? And
Yes.
kind of what I talked about, the Lamarckii, that other Africanized honeybee, is found in the Sam Houston forest. And
Hmm.
they actually tend to be a bit more aggressive than the ones that we know about. And so your bees could be mating with those Lamarckii, and that could be making them more aggressive. So yeah, that's really interesting. Yeah, I wonder, do you hear that the bees tend to be more aggressive in like your part of Texas versus if they're open mating in other parts of Texas?
I hear usually south of us that they're much more
Yeah.
aggressive, down around Dayton and that sort of thing. I think, what do they say? Austin and South is usually more aggressive. But those were aggressive. But then I had one super seeder late this year that requeened. I mean, she was mated probably the last week of October, first week of November, and came back and started laying. I was really kind of sketchy about her, but she's doing great, and they're very calm.
Hmm. Yeah, I wonder. Yeah, it could be different times of the year when they're mating too. So I'm not sure whether the drones are produced from maybe those feral colonies. So those feral colonies could be more Africanized, especially in the southern part of Texas. And maybe they're not producing as many drones then. So maybe if you're mating early in the spring, they'd be mating more with those Africanized drones. So yeah, it's kind of tough to say.
Yeah.
It's one of the things we really don't know where the queens and drones mate at. So we don't know, who your queen's mating with. So that makes a huge difference.
Right. And I will say that hive had drones all winter. There was
Hmm.
drones walking around on the hive in December, late December even.
Oh, really? How many?
Yeah.
You have quite a few?
Yeah, I would say half a dozen
Okay. Yeah,
or more.
here's more about Saskatraz. Yeah, so mostly Italian Carniolans, but a little bit more Africanization. So they have some of those
Mhm.
genetics. So I'm guessing, if they do open mates, that they already have the propensity to be more aggressive, and so they mate with like a more Africanized drone that could make them more aggressive.
Okay.
But yeah, I'm not sure about Buckfast. I've never sequenced a Buckfast. I guess I'd have to go back and see if it's been sequenced before to see.
Yeah. Somebody put up there that Steve Brackman does Buckfast hybrid queen breeding. I thought that he got his from the pole line in Baton Rouge. I thought that's where he got his queens. I listened to his talk this weekend.
C. Brackman, where's he located? I
He's
recognize the
down
name.
around Katy or Fort Bend, I think. Chrissy said something about he does Buckfast hybrid queen.
Okay. Yeah, I'll contact him because if he's willing to give me some of the bees, I can sequence them to see what they look like.
Alvin. Okay. Yeah.
Okay, any other questions where I'm at? Yeah, I think Dodie answered the question by Sue. So what does SMR stand for? It just stands for suppressed mite reproduction. It's primarily the way that bees really sterilize and disrupt reproduction of the mites. This is by uncapping the cells that disrupts mite reproduction and causes them to be sterile. And so SMR is this I guess we've kind of changed that term a little bit. We call them more VSH, but SMR it basically means the same thing. And so Kim said, I heard a talk at the B school that genetics get lost in about 3 generations. Yeah, I'd probably say that's so. I mean, even after 1 generation, you can lose 50% of genetics, especially their open mating. And so it depends on kind of how isolated you are and which drones your queens are mating with, but you can lose those genetics really quickly. And so, I mean, queens are mating with a lot of different drones, like 15 to 20. So, if you buy like a pure Russian or Buckfast or pole line and you're open mating them, then you lose those genetics probably within 3 generations. Yeah.
So people that buy those expensive breeder queens, are they pretty isolated then and controlling their genetics as much as possible?
No, there's no way. Yeah. I mean, in certain parts of Texas you might be. I mean, maybe if you're in the yeah, I'm thinking like southern Texas. What's that big farm that's the size of Rhode Island? Can't think of it. Yeah, but if you're in a huge area where you're isolated and there's nobody around, like a 5 to 10 mile radius, you might be more isolated to make that work. But there's also feral colonies in the forest, so
Yeah.
yeah, I'd say most people aren't isolated enough for it to make a difference. Yeah, but those F1s could be still really good. You still might have 50% of the genetics, but they won't be as good as a pure stock from like a pole line breeding program or a Russian breeding program.
Okay.
So Sandra says that she has 5 Russian hives and she enjoys it and does not find them to be aggressive. Yeah, that's kind of my experience. I think they sort of get a bad rap, and I actually did an aggressive analysis with them versus other bees, and they tend to be a little bit more aggressive, but it wasn't really that bad at all.
And when you say more aggressive, how do you scale that? Is that
Mhm.
okay? They're up because I have a lot of flyers. They fly a lot. I have like half of my apiary. You can wave your hand over them. They just look at you off the frames. They're pretty like lazy Italian, I would say. And then I have half that are they will fly up, but they're not bumping your veil. They're not trying to climb into your sleeves. All of that. They just want to fly around your head a little bit.
Let me show you how I do it. Let me 'cause I presented this yesterday to the apprentice level. Let's pull this up quick. So this is how I do it. It's a scale from 4 to 16, and this is actually just as accurate if you get like sort of a black ball and hit the front of the colony and measure the stings. This is just as accurate and it's really quick to do it. And it's four different traits from scale from 1 to 4. And so one thing I always look at, and this probably only takes like a few minutes, but I look at the flight. So if there's no flight at all, I get that a 1. If there's a few, bees flying, I get that 2. If there's some more, maybe 5 to 10 bees flying around, I get that 3. And if they're actually pursuing you, I get that a 4. For a run, if there's no movements when you open up the colony and they're not moving at all, I get that a 1. If they're kind of walking a little bit across the frame, that's a 2. If they're kind of running off the frame, I get a 3. And then when you open them up and they just like are out the frame within a few seconds, and that's a 4 for me. And then I look at hanging. So if there's no clumping at all at the bottom of the frame, I get that a 1. If there's a kind of one small layer of bees at the bottom, I get that a 2. If there's like a full clump of bees at the bottom, that's a 3. And the bees are clumping and then falling out the clump, I get that a 4. Now for me, I don't wear, gloves or bee suits, so I measure like the stings, and so you don't have to add this if you don't want to. But if I don't get stung at all, that's a 1. If they try to sting you but don't sting you, that's a 2. One sting equals 3, and then more than two sting equals a 4. So I really add this all up and scale from 4 to 16. That's how I sort of quantify it. That's just it's really a simple way to do it. It's actually really accurate. And
What
so
does
they
the
have
hanging have to do with their temperament?
Yeah, typically if you work with Africanized honeybees, they tend to kind of run off and clump at the bottom. So I don't know, have you worked with a really aggressive colony before?
Yeah, where there's a
Yeah.
big clump on the bottom and they just fall
Yeah.
off in a big pile.
Yeah, basically they're running off the frame and then clumping at the bottom of the frame. And so yeah, running and hanging are about the same, but they tend to be more clumping if they're more aggressive.
Okay.
So yeah, if you start seeing the clumps and the clumps are falling off the frame, then that's a sign that they tend to be more aggressive.
Okay. Thank you.
Yeah, that's how I measure it. I'm happy to share this presentation with you guys too, if you want to see how I do my colony inspections. I have it all recorded for the apprentice level.
That'd be great.
So Sue said on inspections, I will occasionally see some dark bees in the hive. It is because they have more of the German bee genetics. My queens are mutts. Yeah, Sue, I'm not quite sure where you get your bees from, but they could be carniolan. So I'd say in general that most of your bees probably aren't the dark German honeybee. You might have a little bit genetics, maybe 10%, but most are probably the carniolan, I'd say. Um, yeah. So I mean we can have you sequence them and see what they are. Be cool. So I need to buy more bees. Okay, so Lee said I need to buy more bees. How do you know for sure what breed you're getting, or you're just getting mutts? Yeah, this is really tough because I know beekeepers market them as Italians. Kim mentioned Buckfast, some say Russians, and really they're mostly mutts. I mean, even Italians. If you actually sequence Italians and Carnioles from California, they're actually very similar. I mean, they're basically mating in the same area, and so they're basically selecting the queen. So Carniole and Italians aren't that different. I mean, they're all a little different in traits, like honey production, things like that. But you really can't know for sure unless you actually sequence them. And so it'd be cool if we can launch a genetic testing for beekeepers to see, what are your bees and are they just mutts? Yeah, and Sue, I will send you. Yeah, I've been kind of delayed with the B school this past weekend, and so I'm finishing the written material, so I'll send that to you afterwards. So yeah, I apologize for not getting that out to you earlier. I've been just kind of getting this B school all figured out. So this all being kind of a written format, so you guys can read and look at it. Okay. Oh yeah, yeah. So Chris said King Ranch, exactly. Yeah, so King Ranch is huge. So if you were managing bees in King Ranch, for example, and I know Blake has his Texas 5000 down there, and so he's pretty isolated. So he'd have a great opportunity to have a breeding program that's isolated. I know some beekeepers have land that's thousands of acres or hundreds and thousands of acres, and so if you have a large enough area, you could be more isolated. Okay, perfect. I'm going to jump to the genetics section now, unless anybody has any other questions. So I always like to start with the breeds and lineages first. I think that's more sort of an easier topic to cover. But I did want to talk about genetics a little bit, and so I'll give you sort of a quick crash course on genetics. Some of you may have had a recent genetics course, or maybe you haven't had one in 20 years, and so I'll kind of walk you through what genetics actually means. And so I know we as beekeepers hear a lot of these different terms like mutation, a marker, a gene, a QTL. And so I'm kind of walk you through like what those terms mean and like how they can be used in breeding and understanding your bees. So as beekeepers, we're interested in a lot of different traits, and we talked a lot about this in the chat. So, we're interested in, honey yield. We're interested in bees that are less aggressive. We want them to be more tolerant to varroa and diseases. And so we're looking for all these different traits. And so when you're thinking about buying, queens or stocks, these are things you should be looking for. And I will say it's really important for you if you are buying stock, just maybe ask the beekeeper where they're getting their bees from. Like, what's their selection protocol? Are they getting from the Russian breeding program, or are they maintaining their own stock? So, I always ask the queen producers, and I'd say in most situations they're probably willing to share some of that information about their breeding program or what they do. And so it's a good idea to know really what traits they're selecting for and what the breeding program they're part of. So we talked about heritable traits, and when you're sort of running a breeding program or a selection program, you're looking for traits that are really heritable because those are easy to select for. And so for the traits that are more heritable, those colonies are more likely to have these traits than other ones. And so, if a beekeeper claims their stocks are real resistance, this is a trait that requires a lot of selection and selection every single year. Because if you don't, you can lose that trait really easily. Whereas if you're looking for propolis, the heritability is really high. So it's harder to lose that trait in a breeding program. And so that's why, I always recommend doing mite washes on all your stocks, even if they claim they're resistant to mites, because it's really hard to maintain that trait. And a lot of beekeepers are buying, Russian stocks or Poline stocks, but they may have gotten it, 4 or 5 generations ago, and those genetics may have been lost, and those traits may have been lost. And so, for a beekeeper that's maintaining these stocks, it's really important to kind of evaluate these and select over, every single year. But if you're purchasing stocks, you might want to evaluate these traits and make sure they actually have them or not. But yeah, the ones that have more heritability, these are the ones that are more likely to stick in your stocks. If you're buying ones that have high propolis, there's probably a good chance that the ones you're buying will have high propolis. But if they're claiming they have, row defense or resistance, that might not be as true because this can be lost really quickly. Okay, so a quick primer on Genetics 101. So I always like to begin here because I know we hear a lot about genetic testing. So, what's the difference between, DNA and mitochondrial DNA? So, honeybees have a, bunch of different cells in their bodies, and each cell has their own DNA. And so you look at the cell, for example, we have the nucleus. Right here inside the nucleus is the DNA. And this encodes every part of the bee and all the traits we're interested in as beekeepers. And so the primary recipe for these traits is coming from this nuclear DNA. But then we have mitochondrial DNA. So this is kind of the powerhouse for the cell. It's in charge of metabolism and energy production. So it has a really important function. But inside of this, it has mitochondrial DNA, so the circular DNA. It's a lot smaller and it's easy to sequence. And so a lot of people have, or a lot of research groups have focused on mitochondrial DNA back, 20, 30 years ago. And so a lot of data that we know about the bees are based on this mitochondrial DNA. Now, what's the problem with mitochondrial DNA? One, it's only inherited by the mother or the queen. So that means if you're looking at genetic diversity based on mitochondrial DNA, it's going to be a lot lower because queen producers are producing maybe tens of thousands of queens from 30 queens. And so that's going to lower the genetic diversity of mitochondrial DNA. And so there's been a lot of research showing that genetic diversity is low in the United States, but the problem is they only look at the mitochondrial DNA, and obviously it's gonna be low the mitochondrial DNA, but the actual DNA that we're interested in actually might be really high. And so this is one thing to consider because a lot of states, and this doesn't include Texas, but a lot of states, they base their Africanization test on the mitochondrial DNA. And mitochondrial DNA is very, very inaccurate. It's not a good way to measure how Africanized a colony is. The best way is to sequence, all 15,000 genes in the DNA and look at the Africanization that way. But, a lot of states and counties rely on mitochondrial DNA, but just know that's not very accurate. So, like I said, they analyze Africanization based on mitochondrial DNA. So Texas, for a long time, started around the 1990s when Africanized honeybees first made it to the United States. They did mitochondrial DNA testing on bees across the state, and they found that Africanization was really high. They stopped this testing around 2007, 2009, I believe. Um, but yeah, they relied on that for testing how Africanized honeybees were. California so really relies on it. So if your bees are really mean and somebody reports that, you have to get tested through mitochondrial testing. And if your bees come back as Africanized, then you have to cull and kill your colonies. Same with Africa, Florida, and New Mexico. Now, the problem with focusing on mitochondrial DNA testing is it's highly inaccurate. So that means it's only accurate 30% of the time. So that means if you have a colony and it comes back as positive for Africanization, there's a good chance that it might not actually be Africanized. And that's one problem with relying on this for states and when they're making regulations, because if your colonies aren't Africanized, but the test comes back as Africanized, you're required by some states to, kill your colonies and really burn your colonies. So Texas doesn't rely on this. They used to do this for Africanization, see how Africanized honeybees are. But we really don't know how Africanized honeybees are in Texas because we're relying on this really inaccurate method for testing. And so I was to kind of begin on that a little bit because, when you hear a lot of papers and reports talking about genetic diversity and Africanization, they really focus on the mitochondrial DNA when they really should be focusing on the nuclear DNA or the DNA actually in the nucleus of the cell. Now, we think about a cell, for example. So each cell has their own DNA. And so some of the DNA can be certain tissues, and they can be regulated in certain ways to produce certain tissues or certain traits. And so based on where the cells are, they play a really important role. So honeybees have this deoxyribonucleic acid, or DNA, and it's a double helix, right? So you have kind of two sets. So this would be an example of a queen and a worker, for example, because they're diploid. So they have this double helix strain. But you think about haploid male, for example, they only have one set of this, so this will kind of be cut in half. But primarily every cell has this DNA. Now for the nuclear DNA, this is a kind of a long linear structure, but for mitochondrial, it's in the circular structure. So for DNA is basically the recipe book for all these proteins and traits. So what happens is this DNA is basically transcribed into what we like to call RNA. RNA is basically the recipe to produce proteins, and proteins are used to make traits. And so, for example, let's say a bee wants to be more hygienic, for example. They make mRNA for that trait, the hygienic trait, and this is transcribed into the protein that goes into the antenna, and that protein allows them to find the dead bees and remove any of the dead bees. And so this is kind of the process where you get the recipe book, and you can imagine DNA as a bunch of different chapters. So imagine DNA is like 23 to 25, well, let's say 16 chapters. Each chapter is a chromosome, and within each chapter is a specific recipe. Now, and one recipe could be for hygienic behavior. So what happens is we take out that one recipe for hygienic behavior. That recipe basically tells you all the things you need to make that recipe, for example, like cookies, for example. So it gives you, hey, maybe we need flour, we need sugar, we need salt, and then it transcribes into the actual cookie or the recipe. And so this is the process how you get from like a full book into a specific recipe like a cookie. So within DNA, we have nucleotides. Now, there's only four nucleotides. We have A, T, G, and C. So even though we have four nucleotides, this is what we use to make up the entire DNA sequence. Now, while four doesn't seem like very much, I mean, you think about just IT and coding, we rely on just zeros and ones for all of our coding, website design, and things like that. And so nucleotides really rely on four. And so even though it doesn't seem like a lot, when you have millions and millions and millions of these can make different recipes and make different types of recipes and different variations in that recipe. Now, here's some important terms that you may hear as beekeepers. You may hear a lot about, mutations or SNPs or SMVs or alleles. These all kind of mean the same thing. We just kind of use different terms. So mutation just means a change in the DNA sequence. So let's say this C changes to a G. That'll be a mutation. Or the C could change to a G, and that could be a SNP or SMV or an allele. So they all mean really the exact same thing. It just means that there's a differences in the nucleotides. Now, I will say that if you're having Russian stock, and if you compare that to Poline or Italian stock, they have about 6 to 8 million mutational differences between them. So if I actually sequence your Italian stock and then compare it to Russian stock, there's about 8 million mutational differences and nucleotide differences between them all. And so that's a lot. And if you have these mutations, you can see how that could impact the traits and how well they perform in your specific environment. So mutation is basically a change in nucleotide. So let's say this gene C changes to an A and T, and if that nucleotide changes, that's a mutation, and that can change. That can either be neutral, so maybe it doesn't do anything, but if it does do something, it could change the entire trait or the behavior of the bees, because these mutations are found in every part of the bee, and these can impact every part of a trait or the behavior of a bee. Now, as you can imagine, like what would a mutation do to a honeybee, right? I mean, it could make them more or less hygienic. We know that mutations in varroa mites make them resistant to a lot of treatments we're using. We know mutations can make bees different colors from black to a goldish color. And so these mutations play a really big role in what we see in as beekeepers. And as you can see, I mean, if we have a really long DNA sequence and we're transcribing this, you can see all the mistakes that can be made. And so even though mistakes are very rare, these mistakes can happen. And these mutations can lead to differences in traits. So maybe, for example, you get a mutation that causes a bee to have a human foot. Now I mean, that could never happen, but there's some mutations that I found really cool in honeybees. So I found mutations where, drones can have different colored eyes. So they can have, black eyes or white eyes or purple eyes as adults. I've seen mutations where, drones actually have two endophalluses. And so I've seen a lot of interesting mutations. Even for you guys may have seen some mutations in your colonies. So maybe some differences in the color of the bees or difference in their eye color or their behavior. And so all your bees have some unique mutations that make your bees different from your other bees. Now we hear a lot about evolution and what evolution means, and evolution can mean a lot of different things, but really what it means is a change in mutation frequency over time. So if you have a mutation in your colony, and maybe that's a really great mutation, so maybe it increases honey production. And because of the increased honey production for that mutation, beekeepers like this mutation. So they select on it. And so that mutation may be in one colony 1 year, but then over 50 years, that mutation spreads to other beekeeper colonies, and that mutation is fixed. So that's basically what it means, is this mutation is, increasing over time. So between colonies, there can be 6 to 8 million mutational differences between new lines. So, if you're a beekeeper and you're buying Russians and you're also buying Italian, just know that one reason that they have differences in these traits is because there's about 6 to 8 million mutational differences between these different stocks or these different lines. And so this is what kind of mutations and how they can evolve in the honeybee industry. So let's say year one. So let's say a beekeeper has this one mutation that makes the queen this dark goldish color. So maybe beforehand, they have 100 colonies and all their queens are this darkish blackish color. But one colony, for some reason, some mutation got into the genome and caused this queen to be a goldish color. And let's say maybe the beekeeper loved that goldish color, and maybe they wanted to select on that goldish color because it made it easier to find the queen. Maybe that goldish color It has some other benefits like honey production, and so they decided to select on that. And so over time, as they're selecting that mutation, the bee slowly becoming transitioning from a darker color to the yellowish color, and then maybe by year 50, 90% of your bees are this goldish color. So that's basically what evolution means, that you have this mutation, and then over time, based on maybe beekeeper selection, or maybe there's some advantage for having that mutation, increases that for your operation or other beekeepers across the US. So when we have a DNA sequence, right, we split those DNA sequences into specific sections, and these are known as genes. And so a gene is basically just a fancy way of saying that this part of the DNA is regulating this one specific protein. So these genes can be different lengths. So some of them can be maybe 100 base pair or 100 nucleotides. Some can be tens of thousands of nucleotides. So they have different sizes. But what they mean is that you basically take out this one specific recipe. So imagine you had a book and each paragraph is a gene. And if you pull out that specific paragraph that has that recipe, that's going to make different types of protein. So for example, gene A or recipe A can make this protein, this specific cool structure. Gene B or that one paragraph in that book makes this structure where it kind of has these kind of four little legs. Gene C may make this protein structure or this recipe where it's these different colors or different types of amino acids. And then we have gene D, which makes this unique structure. And so basically, these are different paragraphs within a book and make these specific recipes or proteins. And so within a honeybee genome, there's about 15,000 genes. And these genes play everything in the honeybee colony. So they play a role in your queen fertility, they play a role in the honey yield, they play a role in their defensive behavior. And so these genes really play a really important role. And one reason that colonies may be more aggressive than other colonies is because maybe this gene A looks like this, so this one's less aggressive, but a mutation happened, and so they look more like this. And that could cause your colonies to be more aggressive than another colony. But yeah, basically these genes are the snippets of the DNA that encode honeybee traits. So proteins are the building blocks for the traits. And so imagine we have a gene for hygienic behavior. And so I'll kind of use this example because I think it's a very clear example. So do you remember the freeze test I showed you on the previous slide where we froze the 300 cells, we put it back into the colony, came back the next day, and removed all those dead bees? One reason the bees can actually find those dead bees and remove them is through their antennas. And so when their antennas have certain proteins, they can actually smell the dead bees and remove them from the cells. And so we know that if the bees are producing these specific proteins in their antenna, they can be more hygienic. But if they're not producing these proteins, they won't be as hygienic. And so this is a really clear example of how this works. So let's say gene A is regulating hygienic behavior. It produces this one protein. This protein gets into antenna and allows them to smell and remove any dead bees. And so that's how we get from the gene level to the protein level to the actual physical trait that we're interested in as beekeepers. So imagine we have a mutation or maybe differences between your colonies. So like I said, there's mutational differences between Italian or Minnesota hygienic. Now, this example of the Minnesota hygienic trait where we froze 300 cells, we came back 24 hours later, and as you can see, this colony is extremely hygienic. There's only maybe 4 cells that are not removed quite yet, and so this colony did a really great job of removing a lot of those dead brood and a lot of those dead bees. But this colony, you can see it's only 66% hygienic. You can see a lot of cells with dead bees are still in there. The bees didn't do a great job of removing them after 24 hours, so this colony isn't very hygienic. Now, one reason that this colony is very hygienic is because it had a fully functional gene. It produces protein that got into the antenna and allowed the bees to find the dead bees and remove it. But for the ones that's less hygienic, they had this one mutation that changed the whole protein structure of this gene. And then that basically stopped the bees from being able to find and remove any of the dead bees. And so this is one way that mutational differences could impact really the trait level and then some of the other traits such as hygienic behavior. So this kind of example, I'll show you, if you find the genetics for this trait. So we know that there's about 126 mutations for hygienic behavior that are associated with it. And so some bees have those 126 mutations and some bees don't have it. If the bees have those 126 mutations, they're more likely to be hygienic. Now, this example where this colony isn't very hygienic, and so it didn't have all those 126 mutations. And so that caused the bees not to produce that protein and to be less hygienic. And by identifying these mutations, we can actually use a genetic test to help beekeepers select and breed for this hygienic test. And here's an example during my time at Purdue University with some collaborators in Canada, and they wanted to compare the field tests to by using just genetic testing. So this is a hygienic score. So as beekeepers, one thing we really want is bees are more hygienic, so nearly 100% hygienic. And this was an uncontrolled stock, so unmated stock. They're not actively selecting for hygienic behavior. And as you can see, after one generation, they're about 75% hygienic. But after three generations, still 75%. Does make sense. It wasn't selected for. Now, for the field-assisted test, they found that if they started after one generation, the colonies were 75% hygienic. After three generations, they became nearly 100% hygienic. And so the field test really works. But by focusing on those 126 mutations, they found the exact same results. So by doing genetic tests, they actually increased hygienic scores from 75% to 100%. So this means one, that's very heritable, but two, that we can find genetic markers or mutations and select traits based on that. And so as we get into the breeding section, we'll talk more about the potential for using genetic testing for breeding, but this really showcases that certain mutational differences can cause a colony to have one trait or another trait. So we hear a lot about QTL, which stands for quantitative trait loci. And that just basically means that there's a lot of genes in one specific area of the DNA associated with a trait. And so, for example, there's usually not just one gene associated with hygienic behavior or stinging behavior. It's usually a lot of genes, and they're usually located next to each other. And so this example of a QTL, so let's say we're looking at stinging behavior. So the bees are more likely to sting you or not. And one reason that they sting you is because they have these four genes that are right next to each other in this QTL. So let's say gene A, gene B, gene C, and gene D. Now for stinging, there's three major QTLs, and we call them, and as scientists, we come up with very creative names. We call them Sting 1, Sting 2, and Sting 3. These are basically QTLs that have somewhere between 10 to 100 genes located next to each other. So one thing we can do as beekeepers is instead of selecting from one gene, we can select for all these genes at once on this quantitative trait loci or QTL. So for stinging behavior and aggressive behavior, there's three QTLs, Sting 1, Sting 2, and Sting 3. And they're located across the honeybee genome. Now, we actually know QTLs for a lot of different traits. We know QTLs for honey production. We know QTLs for grooming. And by identifying these QTLs, it makes it easier to, one, develop a genetic test, and then use the genetic test for breeding and selection. So one thing with DNA is extremely long. So really, so you look at the honeybee DNA in every single cell in a honeybee body. Each DNA strand is about 5 feet long. So I'm 6 feet tall, and the DNA strain is about just as long as me. So it's extremely long and filled with all these nucleotides and these genes. But as you can imagine, that can be very long and hard for the bees to store. So one way that bees and really all mammals and multi-cell organisms do is they actually wrap this DNA around chromosomes. So by wrapping this DNA around chromosomes, it's a really efficient way to store that DNA and store it in every single cell of a honeybee body. And so these chromosomes are basically one way that the bees can store that DNA, even though it's extremely long. Now, we hear a lot about methylation, and methylation is one way to open up and close that chromosome. So as you can imagine, if this DNA is tightly wrapped around this chromosome, it's going to be really hard to read that book. So I guess you can imagine, I mean, let's say you had a 16 chapter book and it has a lock on it. You have the recipes in there, you can make your cookie or your bread recipe, but if you can't open it up, you can't read exactly and read that recipe. That's the exact same with DNA. If it's tightly wrapped around the chromosome, then the bees can't read that recipe and make that specific trait. So one thing they do is they take specific parts of DNA and they open it up so they can actually read that recipe. And that's called methylation. So when they methylate it, for example, that keeps that recipe closed and puts a lock on that recipe so nobody can read it and we can't produce that protein. But if we take that methylation off, that opens up the recipe, we can read it, and we can make that protein. Now, methylation is something that's not heritable by bees, but something that's largely influenced by the environment. So, if your bees are exposed to pesticides or exposed to certain environments, that can cause the DNA to close and also open. So, that's one reason that bees may perform in different environments better than the other ones because they're methylating and opening up that DNA. Now, what does methylation do, and like what type of role does it have? DNA methylation is really important for caste determination. So one reason that queens become queens and workers become queen or workers is because DNA methylation. So because, queens and workers are genetically exactly the same, but one reason a queen becomes a queen is because that methylation opens up that recipe and allows that queen to develop into a queen. So that's one way that the genetic similarities between queen and worker can lead to really huge differences in the reproduction. And so what happens is when a colony is raising a new queen, they're feeding that royal jelly, and that causes those recipes to open up so the queens can get larger, develop larger ovaries, develop the spermatheca. It can also be really important for, some of this nursing behavior. So one reason that bees transition from a nurse to a forager is through this methylation. And so as they're going from a nurse to a forager, that opens up the recipe for the forager and allows them to have these traits and these behaviors of a forager, for example. And so methylation plays a really big role in regulating worker tasks and worker traits. It's also really important for differences in lifespan. So one reason that queens live 2 to 4 years and workers live 21 days is through this methylation. But it's also important for summer bees versus winter bees. So worker bees live 21 days, whereas summer bees live 21 days and winter bees live 6 to 8 months or 2 to 4 months. And one reason for that is methylation. And so, yeah, this is all to say that methylation is largely influenced by the environment. And so your bees could have really great DNA, but based on the environment that your bees are in, that could change a lot of different behaviors and traits of a honeybee colony. Okay, so we talked a lot about genetic diversity, and I always like to touch on, where does genetic diversity actually come from and what plays a role in genetic diversity. So I think I had a graphic here. Yeah. So the last presentation I talked about genetic diversity and how it's actually, really high in honeybees. We've heard a lot of reports about genetic diversity being an issue, but genetic diversity is actually really high, and it could be one reason why we don't see a lot of traits. Now, why is genetic diversity so high in honeybees? There's really four major reasons. So one is, a high recombination rate. So honeybees have the second highest recombination rate, second only to yeast. And so recombination rate is basically one way for the bees to make new variations. So, each bee is a little different. And one reason for that is recombination because it's taking all those markers or those mutations, recombine them in different ways to make basically a new bee. So recombination rate plays a really important role for putting more genetic diversity into honeybees. And that's one reason why I'm never concerned about genetic diversity in honeybees because this recombination rate is so high and they're always adding more genetic diversity into a colony that I don't think it'll ever really be concerned. One reason for high genetic diversity is polyandry. So polyandry just means multiple mating. So, honeybee queen will mate with somewhere between 10 to 20 different males. And by mating with those different males, that adds more genetic diversity into the colony and allows them to be more resistant and tolerant to a lot of these diseases. Mutations. So mutations doesn't add too much, but sometimes your colony may have just a new mutation. So let's say you buy a queen from a source and they have a brand new mutation. That's a new source of genetic variation. And like I said, these bees were imported and managed for, thousands and thousands of years. And over time, there's more and more mutations that get into the honeybees, and that leads to the genetic diversity differences between stocks. And so one reason that, your bees may have 6 million different mutations than other bees is because these mutations have built up and added up over thousands and thousands of years. And then we have admixture. So one thing we do as beekeepers is we try to manage a lot of different stocks and we're making mutts. So by making mutts, we increase genetic diversity. So if we actually compare, bees in Africa or the native ranges in Europe and Asia, they actually have a lower genetic diversity. But here in the United States, we're bringing stocks from across the world, combining them and hybridizing them together, and that increases genetic diversity. And so I don't think genetic diversity will ever be an issue unless you make your colonies really, really inbred, which even if you're doing a closed mating program and instrumental insemination, you probably not do it to the extent where it's going to lower genetic diversity to a concerning issue. Okay, so what is recombination? So recombination is when chromosomes exchange genetic material. And so this is a process called crossover. So let's say you have, this chromosome, what they do is they exchange the red with the blue and the blue with the red, and that produces new combination of genes within the offspring. And so the recombination is happening in all your offspring. And so even though workers can be, kind of similar, one reason that workers are different from each other is because they have different recombinations, combinations of genes. But it's definitely one way to increase genetic diversity within the offspring. And because honeybees have such a high recombination rate, this recombination leads to a extremely high genetic diversity even between workers within a colony. And like I said, they have the second highest recombination rate only to yeast. And if you actually compare recombination rate in livestock or humans, we're really low on the scale compared to honeybees. And it also allows them to have high genetic diversity and adaptability. So this is really one reason that honeybees can live anywhere in the world. So as I talked about the different breeds and lineages, bees can, live in the desert, they can live in mountain ranges, they can live in Russia, they can live in South America. And one reason for that is because its recombination rate is adding genetic diversity and allows them to adapt to their native environments. And we also talk about so it's locally adapted stocks. So if you have your own breeding program, maintain your locally adapted stocks, one reason that your bees can be adapted to maybe West Texas versus East Texas is because of this high recombination rate. So it's adding new combinations of genes, and if you hit that gene right, they can be well adapted to your specific area. Another reason for high genetic diversity is polyandry. So queens mate with multiple males, and this is very, very unique to Apis bees like Apis mellifera. And if you look at bumblebees, for example, bumblebee females only mate with maybe 1 to 2 males. Other solitary bee species maybe mate with multiple males, but only 2 to 3 tops. But honeybees are the only ones that mate with 15 to 20 males. And by doing this, it adds genetic diversity within the entire colony. So if a queen's mating with 15 to 20 males, there could be 10 to 20 of these subfamilies within a honeybee colony. And so this is a sort of an example of this where these colors denote different subfamilies where these red bees have the same father, so they're one subfamily and more related. These green bees have the same father, so they're more related, and so on and so forth. And so we have these different kind of subfamilies within a honeybee colony. And depending on how many queens to mate with, you have more and more subfamilies. Now, what's interesting is, I mean, these drones, these fathers could have certain genetics. And so let's say, the red, for example, these fathers have the defensive genetics. And so these workers or these subfamilies can be more defensive. Maybe in the green, these fathers have more of the honey production traits. And so these tend to be more focused on honey production and so on. And so even within subfamilies, these bees can have different behaviors. They can have different, they can be more aggressive, they can be more docile. So even if you compare different subfamilies within a colony, they can have different behaviors. And we do know that certain subfamilies, they actually may bypass nursing behavior and jump to foraging because they're more bold. So they have these different personalities. So just like humans, right? I mean, I have 2 kids and they have totally different personalities. Same with bees. They have different personalities. And I guess that's not a good example because yeah, I'm the father to both kids, just so you know, but it sort of shows that kids can have different differences in behaviors and things like that. But it's even more extreme in honeybees because they have different fathers. Yeah, maybe I should clear that before I mention that. Um, Another reason for high genetic diversity is admixture. And so I talked about this slide during the previous presentation, but one reason that we have such a high genetic diversity here in the United States is because we're bringing in a lot of stocks, we're mixing them together and making these mutts. So we look at genetic diversity on the Y scale. This tells us how genetic diversity sources are. So the higher the number, the more diverse they are, the lower the number, the lower diversity. So for these different lineages, A, C, M, O, U, and Y, you can see that they have a lower genetic diversity compared to some of these breeding lines here in the United States. And so even the ones that are part of a closed mated inseminated breeding program like Poline and Hilo, they still have really significantly higher genetic diversity. And really the main reason for that is because, we have a lot of bees here in the United States. Beekeepers are shipping bees across the entire United States, and we basically have a bunch of mutts. Okay, so now we have sex determination. So how are sexes determined? So honeybees have a really unique sex determination system. It's called haplodiploidy. What that basically means is based on whether they're haploid or diploid, they become a female or a male. Now, what does that exactly mean? So diploid basically means that they have chromosome sets from both the father and the mother. And so workers and queens, they have a set from the queen and a set of DNA from the drones. And so there's 16 total chromosomes, and this is what diploid looks like for a queen and a worker. But for a male, they're only inheriting half the DNA from the queens. And so as you look at this, they're haploid, but they don't have the two sets of chromosomes. So they're haploid in that way. And so that's one way that they determine whether a bee becomes a female or a male. And the queen actually controls whether they become males or not. And one way they do that is by either fertilizing or not fertilizing the eggs. So if they fertilize the eggs, they get, DNA from both the queen and the drone. They become diploid, but they unfertilize the eggs, they only get genetics from the queen. And so for all the sons of the queen, the sons are actually the queen's actually 50% related to the son, but the son's actually 100% related to the queen. Now, that actually seems kind of funky, but it makes sense because the drones are getting all their DNA from the queen, but the queen's only passing, some of their DNA to the drone actually. So that's so the relationship between the two. So I guess you'd almost think that haplodiploid is a way that things are determined, but it's actually determined by one single gene and one single mutation called the CSD allele or the sex determination allele. And so what happens here is when you're diploid, for example, you have an allele from the mother and allele from the father. Now, when it's a big A and a little b, so they're different, this becomes a diploid female, such as a queen or worker. And so this CSD allele is extremely high in variation. But let's say your things, if your bees are really inbred, or your queen's mating with, like a brother or a son, you could lead to inbreeding on this site. But yeah, just know that if these are heterozygous, meaning that there's a big B and a little b, or a big C and a little c, this becomes a female. For males, they're only receiving one copy from the queen. So that means they're either receiving a big B or a little b. And because of that, they're becoming a male. And so the CSD is really important for becoming male, but it's really based on not just because they're haploid, but because they have one specific allele on that site. Now let's get a situation where maybe this big B male mates with a queen that has a big B. What happens in that situation? We actually get what we like to call diploid males. So these are males that aren't haploid, but they're actually diploid. So they actually have two sets of chromosomes, but they have a big B and a big B right here. So that means they basically are inbred at this point and become diploid males. So this only happens when your colonies are really inbred. And so if your queen's mating with a son or a brother, for example, they can have this inbreeding at the CSD allele and have a big B and little two big Bs. And in that situation, they actually give off a very specific pheromone, and the bees actually start going in them and cannibalizing the diploid males. So it's really hard to actually have those diploid males emerge. I mean, the bees can sense that, and they start cannibalizing or removing any of the diploid males. But this is a situation where you have any inbreeding issues. I rarely see any inbreeding issues. I just some studies looking at diploid versus haploid males and the differences between the two. And so I've done some breeding where I've intentionally made them inbred. But for you guys, you don't have to worry about too much. And if you guys see a spotty brood pattern, that doesn't mean they're inbred. I mean, it potentially could, but there's so many bees out there, and your queens are mating with so many different males that it's probably not an issue. Now, your one question is, what are the differences between a diploid and a haploid male? They're really the same. The diploid males tend to produce less sperm. They don't live as long. They're not as fertile, and so there are some key differences, but they never make it out. They're typically cannibalized before they even become adults because the workers, can smell them and remove them that way. Okay, so what is the relatedness um be- between the bees? So, queens. So let's say a queen lays a haploid male or a drone. The queen is 50% related to that drone, but the son or the drone is 100% related to the queen, right? So that actually makes sense because the drone's getting all their DNA or genetic material from the queen, but the queen's only passing on part of the genetic material to the drone. And then we get into super sisters. So let's say a queen mates with this green male and starts laying future workers or fertilized diploid males. So this male, the father, will be 50% related to the worker, and the queen that laid the eggs will be 50% related to the workers, which makes sense. But because the workers are receiving all the genetic material from the drones, the super sisters are actually related 75%. So you think about like our siblings, for example, we're not 75% related to our siblings, right? We're only, about 25% related. But because so me and my sister, we're 25% related. But if you look at between super sisters within a colony, they're 75% related. So they're more related, they're more willing to take care of each other, and you have these super sisters or super families within a colony. So we think about subfamilies, all those subfamilies are ultra related to each other and what we like to call super sisters. Now, honeybees are polyandrous in that the queen will actually mate with multiple males. So how are the super sisters related to non-super sisters? So let's say a queen mates with this green male and also mates with this orange male. How are the workers related? So let's say these orange are, or these yellow, this green is the super families or super sisters. Like I said, they're 75% related. But for the other subfamily, they're only 25% related. So kind of like, yeah, so 25% related to super sisters. But if there's more workers from this orange, those would also be 75% related. And so because I would just know that, there's subfamilies within the colony and they can be upwards of 75% related to each other, and they're also known as super sisters. Now, let's say there's a queening event and they raise a brand new queen. How related is that? I mean, it's basically exactly the same. The new queen and the sister will be 75% related, but they'll be less related to the worker from the other subfamily. And so you still have a 75% relationship and 25% relationship to this worker. So let's say we have a laying worker. So let's say a situation where you cut out the queen and the queen dies, and your colony becomes hopelessly queenless, and one of your workers begin laying some drones. So that situation is basically similar as a queen, where the workers will be 50% related to their sons, but the sons will be 100% related to the worker or their mother. But then we get a unique situation with the aunts. So this aunt with their nephew will be 37% related, but it's not part of the subfamily. They'll only be 12% related. And so you can get some really funky situations here as you get into it. But really, the only thing you know is that there's subfamilies right here, and they're 75% related to each other, but they're only 25% related to their other sisters. Okay, now we get into genetics and breeding, and we'll cover this in a lot more depth in probably 3 weeks, I think late March. I had to move the breeding section back, but one thing I want to talk about is the potential for genomics and breeding. So one thing we can do as a researcher and help beekeepers is by identifying the genetic markers and using that to breed and select for better bees. And so this is a process called genomic breeding. Genomics stands for the entire DNA that the honeybees have. So all 16 chromosomes, all 15,000 mutations or 15,000 genes, and all 6 to 8 million mutations. So what we do is we look at all those mutations. We find, okay, these 100 are associated with this trait. These thousands are associated with honey production. These 200 mutations are associated with aggressive behavior. And so you find all those mutations, and we basically do a genetic test to breed for some of these traits. And over time, by using genomic selection and selecting bees based on these genetic markers or mutations, we can make progress in any trait we're interested in. So let's say we're interested in honey production. We can do a genetic test, say, okay, these colonies have the mutations for honey production, so we can breed and select for those. Then over time, you'll get this, massive genetic gain. So this is what kind of looks like. So, I mean, as beekeepers, I guess for me personally, these are things I'm interested in. So I want bees that produce a lot of honey. I want bees that have less viruses. I want bees that can be more docile and less aggressive. I want bees that have less pathogens and low varroa infestation levels. And so one thing we do is, we look at these different queens and then identify the mutation. So let's say there's this one mutation, this C, for example. The C is associated with honey production, virus resistance, and mite resistance. Now, these three queens have C, so they're less likely to have these traits. So these queens will have less honey production, to be more aggressive, and be more prone to some varroa. But if these queens have this mutation, so switch from a C to a G, these queens are more associated with high honey production, virus resistance, and mite resistance. And so this is kind of a simple way to show it, but there's hundreds of mutations we can look at, and some of these are more or less associated with some of these traits. So one thing we're doing is developing a genetic test for beekeepers. And this kind of tells us, well, we basically look at the markers and see, okay, your colonies have these markers, so they're more likely to have this trait versus this trait, or your colonies have this marker, and they're more likely to have this trait versus this trait. And so first off, this genetic test will tell us the lineages. So how Africanized are your honeybees, how much O, how much M, and how much C. It also tells you that mixture, so how much of a mutt are your bees. So really the higher the number, the more pure they are, the lower the number, the more of a mutt they are. It also tells you genetic diversity metrics, so how genetic diverse are your stocks. But they give you kind of a breeding score next to each of these different traits. So if you have those mutations for viruses, for example, those would be more likely to have a higher breeding potential for that trait. If it doesn't have those mutations, then it's less likely to have those traits. But they'll kind of combine into a queen score of a 10. So the higher the queen score, it combines all these together. And the higher the queen score, these are the ones you want to breed and select from, and these are the ones that you don't want to breed and select from. And so, yeah, as we develop this test, we're kind of trying to make it more and more accurate. We need to identify the mutations associated with all these different traits, and then form this to a breeding score. And so as a beekeeper, you can look at specific traits. So honey yield, you can see has a breeding score of a 10. So that means this has a higher breeding potential for honey yield and it has the genetic mutations for honey yield. Whereas this one has a 0 for sackbrood virus. So that means this colony is more likely to have sackbrood virus. So this is something we're kind of developing. Hopefully A&M can offer this service in the next few months in the near future, but I think it'll be a really cool genetic test. And it'll be kind of like a dog genetic test or a 23andMe test where you can send your samples into our lab. We can give you some information about the queen scores and whether you're a beekeeper that just wants to know what your bees are, or if you're a queen producer that wants to breed better bees, it can be really valuable. One thing we'd like to develop this for is to help beekeepers verify the stocks that you're purchasing. So say buying nooks or queens from a certain beekeeper, this will allow you to tell you exactly where the bees came from and what type of bees they are. And so are the beekeepers actually market them as buckfast, and are they truly buckfast? Are they marketing them as less aggressive, but maybe they are aggressive? And so this would be a really great way for beekeepers like yourself to identify whether those stocks are actually really, really true or marketed as what they are. Okay, so next week we will cover splitting and diagnosing queen issues. So I was hoping to get the breeding section done next week, but I think we're gonna push it back a few more weeks. So I'd like Lauren, our new program specialist in Angleton, to help me out with that talk. And so next week we'll cover splitting and diagnosing queen issues, and I think it'll be a really good section. So with that, I'm not sure if I have any questions. So Doty shared a video from Keith. Was this associated with polyandry? This polyandry video?
Yeah, yes, it is.
Okay, cool. Yeah, Doty said, yeah, Jones have a grandfather, but not dad. So that's really, really good thing to add. Sarah said, just make sure your queen takes and they don't requeen themselves. Yeah. Otherwise, I don't think I see any other questions unless anybody else has anything else.
Garrett, this is Kim. So on the testing for the Varroa hygienic, like, can you just take like half a frame of brood out of each one of your hives, freeze them for 24 hours, put them back in, wait 24 hours, and then see how much they cleaned it up?
Exactly. Yep. So you can either cut out a part of your frame, or you can put a whole frame in, or use a portion of the frame. But yeah, just put in a regular freezer for 24 hours, or no, put in a regular freezer, let it freeze, put it back into the colony, and then keep it in the colony for at least 24 hours.
Okay.
And if you want to get liquid nitrogen, you can, but I think that's the easiest. That's the easiest way to do it.
Yeah, I think it'd be interesting to do.
Yeah, and I'd say most beekeepers in East Texas have been selected for hygienic behavior, but you can definitely give it a shot to see how hygienic your colonies are. It's pretty easy to do.
Mhm.
So he says, if you believe your hive is Africanized and you replace the queen to get rid of this Africanized trait, would it take 3 generations to get rid of this trait? So it depends how you're replacing your queen. So if you're just grafting off that same colony, so let's say you have a queen that you think is Africanized and you're queening based on that colony, then you still have the genetics in the colony. But let's say you're buying queens from a different source, such as Italian honeybees or Carnioles from California that aren't Africanized, then you can still be queen those and they already made it in California and they won't be Africanized. But if you start with queen cells, just know that your queens are going to mate in that 3 to 5 mile radius and could potentially be mating with drones that have some of those Africanized genetics. So Sarah says, when you're doing a genetic testing on these queens, how are you getting a sample to test? Yeah, so we actually just test the workers. So we don't want beekeepers to sacrifice their queens. And so I know we'll have one available here pretty shortly. Beekeepers will basically just collect basically 10 workers, sending out to the lab, and we'll do genetic testing based on the entire colony. If you want to test your queen, you can, but I don't think there's any beekeepers that want to sacrifice their queen. Okay, perfect. Well, if there's any other questions, please let me know. And so next week we'll cover queen issues and splitting. But yeah, I forget who mentioned it, but we will launch the written material for this lecture. I apologize for taking so long, but I should have it done by the end of this day. And Dan says next week's class 4 hours. No, it's same time from 9 to 12. Is there an issue with the timing of it? Does it say 4 hours? 9 to noon. Okay, so Heather says 9 to noon. So yeah. So Dan, it's only gonna be 9 to 12 next week. Okay, perfect. Thank you everyone, and I'll see you guys all next week.